Okay, so today I'm talking about dihybrids and trihybrids. I'm starting out with the dihybrid. So the first parent is going to have a heterozygous tall and a heterozygous purple flower outcome. Well, that's their genotype. And then the parent two is gonna have hetero tall and a white. And the white trait is recessive, so it has to be homozygous. So first you're gonna do a FOIL method to get the possible outcomes for each child from each parent. So like the possible genotypes, because there's two different traits. So I did that with parent one, and that's gonna be big T, big P, little t, big P, little t, little p, big T, little p. So that's what that ended out to be. And then the next parent is gonna be big T, little p, little t, little p, big T, little p, and then the same thing, little t, big P. So now I'm gonna put in these traits into the dihybrid square. It's a 16 square Punnett square, because it's a 4x4, because it's 4 different possible genotypes crossed with each other. So now I'm gonna fill in the square accordingly. Now when you're filling in the dihybrid, you always wanna put the two the same letters next to each other, or else it's gonna be really, really confusing and really scrambled. Because these are two traits, in order to read them, you have to put the two traits together. So I keep filling it out accordingly. Putting the same letters together, and as I think you can tell by now, is that there's a bunch of genetic diversity here, because there is so many different possible traits, because we're doing two. So now I'm gonna put in the DDDR RDRR. Now the DDDR RDRR represents the different outcomes. So like a dominant first trait, a dominant second trait, a dominant first trait, recessive second trait, recessive first trait, dominant second trait, and then recessive first trait, recessive second trait. So that's what this fills out, rather than being the same letter for each one. That's why RD and DR are two different things. So as I am going through the Punnett square, I am tallying how many times that genotype occurs. So I'm just going through and marking it down how many times each thing occurs. So we have 6 out of 16 for the dominant trait. I think I did this wrong the first time, so I'm gonna go back and fix the math, because I'm like, this doesn't make sense. Oh, wait, no, yes it does. Okay, so there's two different letters because it's two different traits. It's not the same trait, they're two different traits. That's why it's DD and R. DDDRRDRR. That's what that means. So I'm gonna simplify the fraction, even though there's no reason to, but I'm pretty sure that's right. So I don't have to simplify the fraction. So now we are gonna write out the probability for each thing, unless we're not. Yes, we are. Okay, so the 9 out of 16 is gonna be homozygous, and 7 out of 16 is gonna be hetero. And so the dominant trait is that it's a tall purple trait. The dominant recessive trait will be a tall and a white trait. The recessive dominant is short and purple, and then the last one is short and white, because yet again, they're two different traits. So that's the result, that's the dihybrid. Okay, now that we finished the dihybrids, I'm now gonna talk about the trihybrid. So first I'm gonna talk about it's two parents and 3 different traits. That's what the tri is for. So first of all, I'm gonna go over the heterozygous heterozygous. So we're gonna do a tall purple and axial parent first, and then they're all gonna be heterozygous. So then the parent 2 is gonna be the same genotype. So it's all gonna be heterozygous. Both parents, same genotype. So in order to do a trihybrid, the easiest way to do it is Punnett squares first for each trait. So because we're not gonna really do a big square like we did with the dihybrids. So first I'm gonna fill out the Punnett square with the double heterozygous. Now you should know that whenever you're crossing a heterozygous x a heterozygous, it's going to come out with the same outcome, where it's a 1:2:1 ratio. They are all parents are the same. So you only have to really do one Punnett square. So the outcome is all gonna be the exact same. So since we have the 1:2:1 ratio, like I just wrote, it's gonna be the same for those other ones. meaning the final, like the kid outcome will have the same probability as the other ones. So it's going to be a 25% dominant, 50% heterozygous, and then 25% recessive. But that's just with the heterozygous next to heterozygous. Now here's with like a real trihybrid. So the first parent, we have a homozygous, tall, axial, and then a white. And since the white is recessive, yet again, it is going to be homozygous. And the axial is heterozygous. The second one is going to be a, what are we doing? Short, so it's going to be recessive. Then the next one is going to be axial again, that's also hetero. And then the last one I think is purple, I'm pretty sure, yeah. So it's purple, and it's going to be heterozygous or homozygous. Why do I keep writing the E? Homozygous. So now we're going to do the 3 Punnett squares for each trait. So we're going to do the tall x the short. So that's going to be big T, little t is going to be the genetic cross. Nope, that's wrong. I think I changed it. Change the homo tall to hetero. It's not going to be hetero, that's just what's going to happen. So like we did with the last one with the hetero times the hetero, it's going to be the 1 to 2 to 1 ratio. And then lastly, the purple and the white is going to be big P, and like, so it's all going to be heterozygous. That's the result because it's a purebred x purebred. And so that's what we're going to do. Now we're going to do a tree diagram. So we're going to start with the T outcomes. So it's either going to be tall or short. And then we're going to draw the tree lines going out, branching out into axial, or are we doing purple? Nope, we're going to the purple. They're all going to be purple because it's all the same. Even though they're all heterozygous, the dominant trait overpowers the recessive. So they're all going to be purple. Now the other options are going to be axial or terminal. And it's not going to be a 50-50 shot because of the traits that came out from it. So with the T traits, it is a 50-50 shot, but the axial and terminal is a 3/4, 1/4 shot. So now, and I want to find, what do I want to find? Short, purple, axial, I think. Terminal. I want a short purple terminal flower. So now what I'm going to do is I'm going to take since it's short, we're going to multiply 1/2 times 1, but I'm going to simplify it to make 2/4 times 1/4 because of the same common factor. So we get, that's 1/8. That's the probability of getting a short purple terminal flower. Now what if I want a short purple axial tall flower? So instead of doing the probability of finding terminal, we now know that it's still going to be 1/2 times 3/4 instead of 1/2 times 1/4. So 2/4 times 3/4 is 6/16, and that can't be simplified, so the probability is being 6/16. So that's the probability of finding a short purple and axial flower. Now what am I doing now? Simplified it because it's the same base. So okay, this could be a question on the test. Out of 100 flowers, how many are, what do I want? How many are tall, purple, and terminal? So now you're gonna do the probability equation. Since it's the same, we're gonna multiply 1/8 times 100 to equal 12.5. Now I circled 12.5 because that's the technical right answer, but you can't have 12.5 flowers. So it's gonna be 12 flowers. You're just gonna round up to there. Okay, that's it. Those are Dihybrids and Trihybrids.